Okay, we are going to start our discussion on mutations and gene transfer. For this set of slides, I'm going to talk about mutations only. So let's start with of mutations. So by definition, mutation is the change in the nucleotide-based change of a genome. Okay, so it's the change in the gene. It changes in the DNA. Okay, it's in the DNA, not RNA. Usually it's very rare. Because it changes the sequence on the DNA, mutation changes the genotype. All right, and the cause of mutation could be two. One is spontaneous. Spontaneous means it's just error by the machinery of the cells. For example, during DNA replication, DNA polymerase can make mistakes, so that can cause the spontaneous mutation. And the other cause would be induced mutation. So this is caused by something in the environment of the cells. So something that can cause mutations um, is um, is called a mutagen. Okay, so different type of mutagens can cause mutations. The most common type of mutation is point mutations. All right, point mutations mean it affects only one base change. Okay, it affects only one nucleotide. It could be of different types. All right, so you could have substitutions. Substitutions mean one nucleotide is replaced by a different nucleotide. Okay, insertion means a new nucleotide gets added in the sequence, and deletion means removal of one nucleotide. Now, in terms of the substitution mutation, it could be either transition or transversion, depending on the type of nucleotide that gets exchanged. For example, for transition, you have purine to purine, the same type. Okay, it could be A to G or pyrimidine to pyrimidine. While transversion, you go from purine to pyrimidine or pyrimidine to purine. Okay, so transition versus transversion. Transversion means you change the version, okay, like purine to pyrimidine. And then for insertion and deletion, okay, down here, because if you remember, the code in the gene is read in group of three, okay, three nucleotides. So whenever you have a removal of one or addition of one nucleotide, that would result in the shifting of the frame. So the groups of three that's read, that will be different groups of three. Okay, so that means the sequences are going to be changed. The groups of three that will be read because of the deletion or the addition of that one nucleotide will change from the point of the insertion. Okay, again, point mutations could be substitutions where one type of nucleotide is replaced by another one, or insertion, which is addition of one nucleotide, or deletion, which is removal of one nucleotide. Okay, all of those are point mutation affecting only one base change. Talked about, and we're going to talk about the details of the different types of substitution mutations. Okay, so here it is. Again, substitution mutation means you replace one nucleotide with a different one. Okay, but the effect could be different depending on what nucleotide get replaced. Look at the first the sequence on that first DNA strand up here, right? So you have the sequence here. Again, it is read in groups of three, so AAA, ATA, CGT, and GCA. Okay, and when that sequence gets transcribed, you get the sequence UUU, UAU, and so on down here. All right. And then during translation, it's going to be translated to phenylalanine, tyrosine, alanine, and arginine. Okay. These are the amino acids that get translated from the codons in the mRNA. Now, let's assume that we have a mutation on the third nucleotide right here. So from AAA, it becomes AAG. So when it is transcribed, it becomes UUC. Okay. If you remember, when we look at the genetic code table, okay, sometimes you have one amino acid that can be coded by different codons. All right. So this is the case where that happens. Okay, which means that UUU codes for phenylalanine. When it's mutated to UUC, it still codes for phenylalanine. So if I ask you, is there a mutation here? Yes, because there's a change in the nucleotide sequence on the DNA, right, from AAA to AAG. But is there a phenotype change? Okay, the product. Is there any change in the product? No. You see here, phenylalanine, same phenylalanine. This is called silent mutation. Okay, silent mutation. So there's a mutation in the DNA sequence, but there's no change in the product and the amino acid sequence. So this is called silent mutation. And then the second one is a missense mutation. Okay, missense mutation. So for example, this one here, you have the change in the third triplet here. Okay, so from CGT becomes CCT. So CCT is transcribed to GGA, right? So here, uh, the original sequence transcribed to GCA, it gives you alanine. In here, when it's replaced to GGA codon, it is translated to glycine. Okay, glycine. So it still makes sense. It is still made into amino acid. It's still translated. Okay, but it makes a different sense, a different amino acid because originally it's alanine. Now you have glycine. Okay, so this is called missense. All right, missense. That means it still makes sense, but it's a different sense. Okay, that is because the amino acid has changed. Okay, so this is called missense. Now the third one, you can see here, it happens on the second triplet. So from ATA it becomes ATT. And when this is transcribed, you get UAA. And if you get to that codon table, UAA codes for stop codon. Okay, so what happens then when you get to after that mutation occurs? It's gonna stop right there. Okay, so the protein that you get is actually shorter. You get truncated protein. All right, so that's because the stop codon is not recognized by any tRNA, right, during translation. So this is called nonsense. Okay, because stop codon there, it does not make sense. Okay, it stops the translation. So those are three different types of substitution mutation. All of these are substitution, meaning you have a nucleotide that gets substituted with a different one. Okay, but the effect could be either silent, meaning there's no change in the amino acid sequence, or missense, where you get change in the in the amino acid sequence, or nonsense if it becomes a stop codon. Okay, so it stopped the translation right there. All right. Everything after the point of insertion, especially if this happens in the beginning of the sequence, right? So everything behind it is going to be affected. The second one here, you have deletion. Okay, so you have removal of one T, and that is this T right here. Okay, and the effect again is the same because you have the shift in the reading frame. So instead of ATA, now you have AAC. Okay, so taking the next one, which was on the third triplet right here, it becomes the second one. and so on. Okay, again, it's the shift in the reading frame, and the effect is going to be the same here. All right, whether it's insertion of one nucleotide or deletion of one nucleotide, the effect is going to be the shift in the reading frame. Okay, and it's going to change all the sequences after the insertion point. Okay, except for there may be some silent mutation there, but most likely that all of those amino acids are going to change. All right. Next, we're going to talk about mutagens. Okay, this is important for us because this is how different things can cause mutations, right? So chemicals and different things like that. So all of different chemicals or treatment that can cause mutation, these are called mutagens. So one type of mutagens is radiation. Okay, radiation. If you remember, radiation is part of this electromagnetic spectrum that includes the light and so on. So we can divide radiation that causes mutations into two subgroups. One is called ionizing radiation, the other one is non-ionizing. Okay, so ionizing radiation includes X-rays, gamma rays. These are um, the radiation that has very high energy. Okay, because their wavelength is very short. Remember this, you have this inverse relationship between wavelength and energy. So these are the one that have very high energy, so they can ionize. Ionize means you create ions. You actually you can remove electrons from atoms. Okay, so they become ions. So the effect is pretty bad. Okay, so with X-rays and gamma rays, what happens is the chromosomes can break. Okay, because it's very high energy. Now, on the other hand, you have non-ionizing radiation. An example of this is UV. So if you remember, UV is right here, okay, compared to this, it has lower energy, okay, because it has higher wavelength. So UV has less energy than the ionizing radiation, so it does not cause ionization, okay, so that's why it's called non-ionizing radiation. But it has enough energy to cause something If you remember, we talked about this in the lab. This is called pyrimidine dimers. Okay, in the presence of UV light, there's enough energy to cause the formation of the thymine dimer, that is, or pyrimidine dimers, either thymine or cytosine that are next to each other. All right, and so this can cause eventually when you have the bulge because of the formation of the thymine dimer, um, the DNA polymerase is going to read this incorrectly, and then during DNA replication, it's going to create a change, okay, mutation in the sequence of DNA. All right, so again, mutagens could be either in radiation, could be ionizing, which includes X-ray and gamma rays. They cause break in chromosome, and then the other one is non-ionizing radiation, which is UV light, and that can induce pyrimidine dimers. Three different types. Okay, one is analog. Analog has analogs contain different chemicals that have similar structures as the actual nucleotide. Okay, so they compete with the normal nucleotide because since this is like competition because they have similar shape. And then the other one is chemicals that can change the actual nucleotide. Okay, so if, for example, here nitrous acid can convert A to G. Okay, so it removes some of the atoms there, so it converts A to G. So it changes the nucleotide directly. And then the frameshift mutagens such as ethidium bromide, acridine dye, they insert into DNA. So this causes frameshift mutagens because it shifts the frame again, okay, because it inserts itself in the DNA. So now the DNA polymerase is going to read the sequence again incorrectly because it looks like it's inserting itself. It's just like an insertion mutation, okay. So because of the bulge there, DNA polymerase can delete or add bases around the bulge. Okay, so let's look at um, the description here. This is an example of nucleotide analog. Okay, so look at this. This is structure of thymine or the T base of a DNA, and this is bromouracil. You see how similar the structure is, except for the brom atom here in place of methyl group. Okay, so when this chemical is around during DNA replication, this is going to be um, it's going to be recognized as thymine. Okay, DNA polymerase is going to think that this is thymine, so it gets incorporated right here in place of T. Okay, and that causes because this Bu, the bromouracil, actually pairs with G instead of with an A. Okay, so you see here, originally you have TA, and here it becomes G, right? Bu and G, and then in the next replication cycle, this becomes C and G. Okay, so TA becomes CG. So that's what happened when you have nucleotide analog, right? Um, for example, this is a chemical that can insert itself in between nucleotides. Okay, you see here? Okay, so the shape is just right so that it can insert in between nucleotides, and the result of it is the formation of this bulge here. And so when DNA polymerase tries to read the sequence during DNA replication, it's going to either delete or insert more nucleotides. Okay, so what happens here is you have frame shift. It's a shift in the reading frame. Now let's look at the DNA repair. Okay, pyrimidine dimer. We talk about this in the lab. Um, there are two different types of repair for pyrimidine dimer. One is, remember in pyrimidine dimer you have the T and T that bonded together. They're side by side instead of they're pairing to the opposite strand. Okay, A on the opposite strand. Now the repair of this is two ways. One is the light repair in the presence of light. Okay, actually this bond is broken. Okay, so this reverse of the formation of the pyrimidine dimer. Now you get it back normal. All right. And the other one is the dark repair. This is in the absence or in the presence of light, but it does not require light. With this type of repair, the part that has the bulge, okay, the dimer, actually it gets cut out. Okay, so there's a fragment that is a section that includes the dimer here that gets cut out, removed, and then DNA polymerase 1 is going to refill the gap, and ligase is going to connect the fragments. Okay, so again, pyrimidine dimer can be repaired two ways. One is light repair and one is dark repair. Light repair just reverse the formation of the thymine dimer, and the dark repair actually removes a segment that includes the dimer and then replacing it the correct sequence. One is this is when what happened when DNA polymerase makes mistakes. Okay, so here instead of a T you have AG. Okay, um, so this repair is called base excision repair. Okay, so the base that was wrong is going to be excised, removed, and then it's going to be replaced again by DNA polymerase 1 and ligase. Now the other one, yeah, this is called mismatch repair right here, mismatch repair. So base excision repair occurs during DNA replication. Okay, it gets fixed right away. Mismatch repair occurs after DNA replication, and again you have mistake here. What happens is this is similar to what we saw before. This the part that has the mutation, it's a segment. All right, the segment right here get removed, and then that is replaced by the correct nucleotides by DNA polymerase 3. Okay, so those are the two types of DNA repair. All right,